Okay, I'll go ahead and get started to keep things on time. Today I want to talk about the process of speciation within the context of mitonuclear coadaptation. And so mitonuclear coadaptation is a necessary outcome of the genetic architecture of eukaryotes. So in eukaryotes, the electron transport system is encoded by two genomes. The parts that come together to form this machinery are both nuclear encoded and mitochondrial encoded. And so that means that these components have to fit together and function together properly, or you get reduced respiration and reduced fitness. So we can define mitonuclear coadaptation as the coordination of function by the products of the mitochondrial genome and the products of the nuclear genome towards an optimal oxidative phosphorylation. And we would define optimal OXPHOS for each species, depending on its life history and the climate, what have you, that it lives in. Okay, now a key point is that we can only maintain mitonuclear coadaptation through perpetual coevolution. And the reason for this is these are not static genomes. And in particular, the mitochondrial genome is prone to rapid evolution because, in general, it mutates at a much higher rate than the nuclear genome. And in general, there's no recombination. It's a haploid genome that's transmitted without recombination. So high mutation rate and no recombination, you get a lot of changes. And some of these changes are functional. And so there has to be coordinated changes in the nuclear genome to maintain mitonuclear coadaptation, which is core to organism fitness. So those are general processes of eukaryotes. And for the rest of the talk now, I'm going to shift to speciation, and I'm going to focus on my favorite group of eukaryotes, which are birds. And for a number of reasons, birds are probably the best group of eukaryotes in which to understand the process of speciation. I think for birds, maybe things are a little less confusing than for other groups. And I can give you rationale for that. But there's been books written just on bird speciation, and that's almost unique among eukaryotic taxa. And so if you start investigating bird speciation and mitochondrial processes, you instantly run into the literature on DNA barcode. And so this really isn't a literature that's connected directly to speciation theory. DNA barcoding was created by taxonomists as a way to diagnose taxa with DNA sequences. And so, but what these guys discovered about 2 decades ago was that a mitochondrial gene, and actually a portion of a mitochondrial gene, was really good at diagnosing species. And so, and what I mean by good at diagnosing species is, and it works really well for birds, is there's consistently a barcode gap just at the place where taxonomists had already defined a species gap. And so a barcode gap is a significant change in mitochondrial sequence that would be in contrast to the relative similarity of DNA sequences within a population that we define as a species. And so DNA barcoding has been really successful in birds. And one of the ways we assess the effectiveness of it is to compare the species that are identified with mitochondrial sequence to the species that have been identified by taxonomists. And in birds, it's always better than 93% match between what taxonomists have described as species and what a DNA sequence describes as a species. Now, sometimes this is stated as DNA barcoding works 93 or 90%, 95% of the time. But that statement would assume that taxonomy is perfect and all the error is on the DNA barcoding side. And we know taxonomies aren't perfect. And so there's some error on the barcode side and some error on the taxonomy side, but it's a really good match. Now this was shown most effectively, most convincingly, in a study by Tavares and Baker in 2008, in which they went to GenBank and extracted all the DNA sequence, the mitochondrial sequence data that were available, and they looked for pairs of sister taxa. And they found 60 of these pairs. And so then they went through and they looked to see where DNA barcode sequence would cluster species compared to where taxonomists had drawn species boundary. And in 60 out of 60 of these cases, the mitochondrial clustering matched the already defined species boundary of these groups. So this is really a striking observation that avian species have unique mitochondrial haplotypes. Now, there's a number of explanations for this, but I want to consider that this could be a product of the necessity for mitonuclear coadaptation maintained through coevolution. And we can make this point most effectively with just a simple descriptive model of allopatric speciation, just like we would teach in sophomore evolution. But now we're going to keep track of both the mitochondrial genome, which I just showed as a little circular chromosome, and then in particular, these would be nuclear chromosomes, but we're going to focus particularly on that small subset of nuclear genes that interact directly with the mitochondrial gene. So it's only a very small portion of the entire nuclear genome. Okay, so this is a simple model of allopatric speciation. So we start with a panmictic population with no structure. Something happens to divide the population into two parts that no longer exchange genes. So these are now daughter taxa on unique evolutionary trajectories. And over evolutionary time, inevitably, statistically inevitably, the changes are going to accumulate first and foremost within the mitochondrial DNA because it has a much higher mutation rate, and in birds, it's non-recombining. And so we know this. This is why so much avian systematics is based on mitochondrial DNA sequence. But we got to remember we have to maintain mitonuclear coadaptation. It's essential to organism fitness. So as the mitochondrial genome changes, some of the changes are going to be functional. And in order to maintain mitonuclear coadaptation, there's going to have to be matching changes in the nuclear genome. You're going to get coordinated coevolution of that set of interacting nuclear and mitochondrial genes. So as we go forward in evolutionary time, we may hit a point at which these populations are back into contact again. And what can happen here is that even if there's been insufficient divergence in the nuclear genotype at large to stop gene flow, these specific coadapted sets of mitonuclear genes can create dysfunction in hybrids. If matching a blue-coded nucleotype with a green-coded, mitochondrial type with a green-coded nucleotype creates dysfunction, then we won't have gene flow across the populations. The mitonuclear incompatibility would disrupt gene flow. So we can state this as a species concept. We can state it as a mitonuclear species concept where a species is a population that's genetically isolated from other populations specifically by these incompatibilities in mitochondrial and NMT genes. That's the nuclear genes that go over and work in the mitochondria in close association with those mitochondrial genes. Now, this process can work perfectly well with post-zygotic sorting. Low hybrid function, so hybrids have low survival and there's no transmission of genes across the species boundary. But if you're an individual mating within the system, it's very much to your advantage not to walk into that. It's very much to your advantage to recognize the genotypes that will give you fit offspring and not lose your fitness to an unlucky choice. So we can imagine, and this is just the concept of reinforcement. We just heard about it in the last talk. It's a basic concept in evolutionary biology. So we can imagine for birds something as simple as black wings versus red wings can allow a choosing individual to sort correctly in a pre-zygotic mate choice. So if we go back to that Tavares and Baker paper and simply show the appearance of the individuals in those populations, we see a striking fact that the DNA barcode gap falls exactly where there's also an ornamentation gap. In 60 out of 60 pairs, it's either a song gap or, in most cases, a plumage gap. So we go from gray-bodied phalarope to red-bodied phalarope. I particularly like the golden eyes they almost are wearing a barcode. So you go from mostly white bird with a little black to a mostly black bird with a little white. You can imagine these are very simple patterns for species recognition, for sorting correctly. For this to work, though, we have to have linkages. In order for this to work, what we're proposing here is that a choosing individual wants a particular genotype, and in order to do that, she can't see the genotype, but she can see the signal of species identity, the ornament, the song, or the plumage that would indicate type if it's linked. So we need linkages between those specific nuclear genes that interact with mitochondrial genes, the ornamentation genes, and if mate choice is genetically determined, it may be imprinting, which actually works fine. But if it's genetically determined, that should also be in this linkage group. Because in that way, by choosing plumage, the female is choosing a genotype that matches her genotype. The goal is to match blue mitochondrial type to blue nuclear type and red mitochondrial type to red nuclear type. You can do this through choice of ornamentation if there's the correct linkages. Okay, is there any evidence to support any of this stuff I'm telling you about? Well, there's a growing body of evidence in the avian literature that's striking in the patterns that are coming forth. So in numerous bird species now, and I just pulled down little illustrations of some of the recent ones that have appeared in the literature. There's a huge growing literature using modern genomic analysis to assess genetic structure between closely related bird species. What the emerging pattern that's being seen again and again now in these sorts of avian analyses is that autosomal nuclear genes tend to diffuse across species boundaries. We get blurry boundaries between species with autosomal nuclear genes, but we get tight boundaries with mitochondrial genotypes and Z-linked genotypes. The mitochondrial genotype is why barcodes work. If we didn't have tight boundaries with mitochondria, we wouldn't have a DNA barcode signal. But also Z-linked genes work. The best data come from pied flycatchers, from a number of European labs, but especially Sater's lab. These are two closely related songbirds in Europe. And using not modern genomics and not mapping particular genes, but old animal breeding models, because they got pedigrees for all these things, they've been able to show clearly that genes for ornamentation that are used in species recognition, genes for mate preference are Z-linked, and so are incompatibility factors. These are genes that are determining hybrid dysfunction. They don't know what these genes are, but they can show through breeding studies that they're Z-linked. I'll bet the farm that these, at this point, unknown incompatibility factors are those nuclear mitochondrial genes that are going to interact specifically with the mitochondrial genes to create respiratory function. So we can illustrate this with a simple graph. What we're seeing now in bird taxa, especially in the pied flycatcher, is a loose boundary with autosomal nuclear genes, but this tight discord with mitochondrial genes. That's why we get a DNA barcode gap. With plumage genes, that's why field guides work. We call them binoculars. I never see birds that don't fall into one category or another, and I'm a fanatical bird watcher. You get these tight boundaries. Z-linked genes now are turning out to have tight boundaries as well. So this is an unfolding story. I think it's going to be really interesting as we start to actually map some of these genes. So I'll stop there and have time maybe for one question. Yes.
Question. I'm wondering, how do you calculate the limiting barcode gap, because there are
Yeah, there's a lot of ways. The Tavares and Baker paper did it the best. They did it just by, they used a cluster algorithm in which they simply looked for monophyly in their group. So they didn't use a 2% or a 10%. They simply looked for where you get monophyletic clustering within the group. So that's not the kind of work I do, but I've seen talks at this meeting where there's some new Bayesian models for clustering. There's a lot of ways to do that, but you tend to get really clear signals in the mitochondrial DNA sequence. And it doesn't have to be CO1 or COX1. The other mitochondrial genes have a big signal too.
And I was thinking that you would see anything similar, I think, research on
No, it's a mitochondrial phenomenon as far as I can tell. Yeah, there is a lot of interesting plasmid stuff, but it's different. Mitonuclear is really fundamental because it's all eukaryotes, and it's really at the base of eukaryotic evolution. It merges That's why speciation is fundamental to eukaryotes, because it's what defines eukaryotes, minor nuclear. Okay.